Hi everyone, Dr. Hinkie here. Welcome to our first video lecture. You'll hear me say again and again and again throughout this term that you should break down the chapters into bite-sized pieces. I am going to do the same thing with the lecture videos. So I'm in the process of re-recording my entire chapter all at once, 60 to 90 minute long videos into section by section videos. And that's how you should tackle the chapters, one section at a time, focusing on the learning objectives for that section, reading to understand those objectives and to achieve those, and then stop. Let your brain process that information for a little bit before moving on. Our learning in this class is progressive. That means we have to build on earlier concepts as we go along. So if you read to rush through just to check it off the list, I read that chapter, but you don't take time to really ponder and consider it and understand it, you won't be able to build on those future concepts. So it's worth breaking down, spending a little bit of time. Do take a look at our weekly success tip. There's a pre-class weekly success tip on 5 study strategies that nobody learned in school, but they were really effective and important to use, especially in higher education, because now you're being asked to think and learn in new ways, more complex ways that aren't necessarily required through elementary, middle, and high school. So do take a look at those, and I will try to set things up and proceed through our class in a way that utilizes those effective learning mechanisms. So moving on to our first chapter. To introduce us to the invisible world that we'll be studying this term. And section 1.1 is what our ancestors knew. So we're really getting some background. So in this chapter, if you really feel like, oh my goodness, I don't know any of this, this is introducing just broadly. Here are the things we're going to go into more depth on through the term. So understand the material generally, because that's what this is, a big, broad, general overview. And these are the things that we're going to get down to the nitty-gritty on as we move through. So our objectives for this chapter are to describe how our ancestors improved food with the use of invisible microbes, describe how the causes of sickness and disease were explained in ancient times prior to the invention of the microscope, and are discovering that there are living organisms responsible for disease that are smaller than the naked eye can see. And describe key historical events associated with the birth of microbiology, including contributions by Antony van Leeuwenhoek, Louis Pasteur, and Robert Koch. So first, what is microbiology? Microbiology is a specialized area of biology that deals with tiny life forms called microbes or microorganisms. So micro, small, biology, study of living things. So we're studying small living things. We need microscopes, sometimes light microscopes, sometimes even more powerful ones to see them. Microbes are ubiquitous. That word ubiquitous means they are found everywhere, everywhere on Earth. They've adapted to a wide range of conditions, from the very deepest of the deep sea and the high pressures that exist there that you have to accommodate, to frozen tundra, even Antarctica, the Antarctic, with its freezing temperatures, the high salinity of the Great Salt Lakes. Every habitat on Earth is colonized by bacteria. So they've been around for over 3.5 billion years. It's the longest living life forms on Earth, throughout the evolution from those very simple single-celled life forms through to us. Evolution is the accumulation of changes that occur in organisms as their populations adapt to their environment. And we'll get into that a little bit more when we talk about genetics and how those changes accumulate. Tiny changes in our genomes accumulate over years, centuries, generations, generation after generation, those changes. Some organisms, because of those variations in their genome, are better able to survive and thrive and pass on their particular set of genes, adaptations that make them best able to survive in the environment in which they live. So our early understanding of microbes, humans have been using microbes for much longer than they could see them. So just through observation of things that happened in the world, probably through accident in many cases, humans discovered that they could manipulate factors in their environment and take advantage of those. And what they were manipulating was the outcome of metabolic processes from microbes that they could not see. There's evidence that humans were using fermentation to preserve foods as a preservative and to enhance taste. And there's evidence all the way back to Neolithic China as early as 7000 BC. Humans have used microbes to make fermented foods like beer and bread and cheese and wine throughout history. When we talk about fermentation, fermentation is a metabolic process that allows organisms to get energy, ATP. Remember, ATP is the form of energy that cells can use. And so organisms have to break down the chemical bonds in their food, release that energy, convert it to ATP, so that they have energy that their cells can use. Aerobic cellular respiration is how we do that. If we do go anaerobic, if you're doing a fast sprint workout where you can't get enough oxygen to sustain the needs of energy of your cells, we will convert to lactic acid fermentation. Our cells, our muscle cells, are able to use fermentation anaerobically. Fermentation does not require oxygen for short periods of time. The end products of fermentation are ethanol, or alcohol, and carbon dioxide for some organisms. In our cells, the outcome is lactic acid. So fermentation can produce acids as an end product, or alcohol, and sometimes carbon dioxide. We see that when we make bread. So yeast is a single-celled organism, a fungus, that's able to use fermentation. It's a way to get energy when there's no oxygen available. So we put that yeast in the dough. The dough has flour and water. That yeast becomes activated, begins to break down the flour into its component glucose molecules, simple sugars, and then ferment those. When it ferments, it breaks the sugar down into pyruvate and then converts that to ethanol alcohol and releases carbon dioxide. That's the reason the dough rises. That carbon dioxide, those gas bubbles, are captured in that dough. And if you've ever driven by a brewery and a bakery, they smell the same because baking the bread burns off the alcohol that's the result of fermentation. So humans have been taking advantage of that fermentation process for well before the time they could actually see microbes with a microscope. As far back as more than 3,000 years BC, there's evidence that humans were using microbes to treat infections. So Ötzi the Iceman, from between 3350 and 3105 BC sometime, he was discovered in the Alps on the Austrian-Italian border in 1991. So this 5,300-year-old body was frozen in the ice. So it was very well preserved. His clothing was preserved. Much of his clothing, some of his food, evidence of how he died. He is older. Put him in some timeline perspective. He's older than the Egyptian pyramids, older than Stonehenge. So Since prehistoric times, there's evidence that humans have used microbes both in the treatment of disease and in the preservation of food. So in Etsy, who was frozen, there were signs of parasitic infections and also signs that he was attempting to treat those infections with fungus and herbs. There's a link in here if you go into the PowerPoint and play that in full screen mode and get some more information on Etsy here. More recently, in medieval times, there have been many different attributions of what causes disease. A lot of it due to superstition, malhumors, something evil in the air that was inhaled, punishment from God, lots of different reasons. But even with those attributions of supernatural forces as the cause to disease, there seemed to be some understanding at a basic level that disease could be transmitted through things that couldn't be seen. And so that transmission could be controlled. And that included through quarantine. We see with leprosy and leper houses, and they would put lepers into quarantine to keep them away, keep them from transmitting that to other people. The ancient Greeks and the ancient Romans understood that disease could be transmitted and spread. So the Romans actually built aqueducts to protect from waterborne illnesses. They had the first sewer system. So the Cloaca Maxima, or the greatest sewer, was a waterway, a man-made trench waterway that ran through Rome, this whole length here, to carry wastewater from public latrines, public outhouses latrines that emptied into the Cloaca Maxima and carried that wastewater to the Tiber River. In ancient Greece, the Greeks made great strides toward understanding the invisible forces, those microbes that caused infection. Hippocrates is considered the father of Western medicine. He was first to propose that there are living organisms that are responsible for disease. Thucydides is considered the father of scientific history. He was actually a historian in ancient Greece, and he was an advocate of evidence-based analysis and cause and effect reasoning. We'll talk more about Robert Koch, and he is considered the first in modern history to establish this cause and effect rather than just correlation with microbes. But he had the benefit of having a microscope. Thucydides did not have that, but just through observation and gathering of evidence, he realized that after the Athenian plague, which he survived, that he and the other survivors, never got reinfected. She was the first to propose that there was some mechanism of immunity. So there was some understanding here without really having all the information because they were unable to observe microscopic organisms. So then we get to the golden age of discovery in medicine. And first, one of the greatest characters in microbiology, I think, is Antony van Leeuwenhoek. And look at these dates, 1632 to 1723 is when he lived. So it was still a while ago, not a whole lot of understanding of microbes. He's often credited or comes across being the person who invented the microscope. He did not. Microscopes already existed. Scientists were well underway to exploring the living world, and they did have some basic microscopes. The Royal Academy scientists belonged to academies, so each country tended to have royal academies. The Royal or a scientific academy. There were Royal Academy in London, in England, in Italy, in Rome. So there are a lot of scientists working. Van Leeuwenhoek was not a scientist. He was a draper. The family business was clothing, was um, making draper, but working with cloth, weaving cloth. And as a cloth merchant, he was very interested in looking closely at the threads and the weave. And so, as a hobby, he started polishing glass to make lenses that gave him ever increasing ability to see finer and finer grain scale on these weaves of the cloth. It was a hobby for him. But through his hobby, he developed a more powerful microscope than anything that the Royal Academy had. So the interesting part of this is he would write letters. The way scientific information is communicated back then was through letters and publishing and writing to other scientists about your discoveries, your observations, your experiments, and sharing this information that way. But there was something of a formal format to follow and structure, but Van Leeuwenhoek was not trained as a scientist. So he would just write letters like you would write to Aunt Tilly, and he would write very, hey, how are you, pal, what's up? Very casual letters. He would talk about the gossip in the neighborhood and then tell them about, hey, I have this lens that I developed, this microscope, and I'm seeing these wee beasties. He called them animalcules, animal molecules. And it turns out with his simple single lens microscope, he was able to see bacteria and protozoans, single-celled organisms. It was the most powerful microscope to date. This is what it looked like actually. Put the sample here on the tip of this needle and look through this little tiny lens. So there are actually two metal sheets with the glass lens pressed in between them would look through. And you would use these screws here to move about your sample into focus. Trains going by outside, probably hear that now. So a very simple microscope, a single lens, but he managed to polish this lens in a way that made it more powerful than anything the Royal Academy had. So after years of writing to the Royal Academy and these scientists saying this crazy man up in the Netherlands, he's not a scientist. What is he? He couldn't possibly have a better microscope than ours. And finally, after just continuing and being persistent and writing these letters and saying, you should see the stuff I'm seeing here, they sent some representatives, and sure enough, his microscopes, and by now he had just made many of these because it was his hobby, and his microscopes were far more powerful than anything they had. They offered to buy them from him, and he said, no, they're mine. So they were his toys, his inventions, he kept them. Eventually, the Royal Academy did issue him an invitation to become a member, and he was able to pursue scientific studies using his microscopes. He observed everything from drops of rainwater to see the microbes in there, to the plaque scraped from his own teeth. There's some other information here. Funny story. He was, at this point, people didn't understand reproduction, and they did not understand that humans produced eggs, females produced eggs, males produced sperm. And someone from the Royal Academy did ask him, hey, look at this. And so he was the first person to observe sperm under the microscope. And he was so disgusted by the thought. He's the first person to see sperm, to see these living cells with flagella moving. And, he said, please don't publish this. Please don't let anybody know I did this. He was totally embarrassed by it. But he was the first person to actually show that these cells were moving and part of reproduction. They didn't make that connection quite then, but it's just an interesting, funny side story about Van Leeuwenhoek. Don't read the textbooks very often. He was a very interesting character. So Antony van Leeuwenhoek, he's considered the father of microbiology because he's the first one to see microbes with his microscope. Louis Pasteur, and look at the dates here. So we had a microscope in late 1600s. It took a couple hundred years before we really started to understand what those microbes were doing. Louis Pasteur was the first to demonstrate that fermentation, this process, we've been using it for millennia to make alcohol, to ferment foods, to preserve them, was caused by microbes, by single-celled organisms, bacteria and fungus primarily. He also developed the process of pasteurization as a way to kill microorganisms that cause spoilage. We'll look more at pasteurization. It uses heat, but not such a high heat that it can change the texture or the flavor, the taste, the quality of foodstuffs, of dairy in particular. So it will reduce the load, the number of microbes. It won't sterilize the liquids or the food. It doesn't kill all of them. That would require higher temperatures and pressures, and it would destroy the taste and texture of the food. But we still use pasteurization to this day. He also developed vaccines for the treatment of a number of different diseases, including rabies. And as we'll learn when we get to our chapter on cells, he disproved the hypothesis of spontaneous generation, meaning life arose spontaneously from nothing. Robert Koch is another interesting character we will get into. We'll see him in future chapters, but especially we'll take a look more closely at Koch's postulates when we talk about epidemiology in chapter 16. And as I mentioned earlier, we talked about Thucydides. Koch was the first to use this scientific process to demonstrate a connection between a single specific isolated microbe and a known disease. So this microbe causes this disease. He established that causation. So he was really our father of etiology. Etiology is the study of the cause of disease. And he also went further. He identified a number of specific diseases, and the process he used, he laid out into a series of steps that we call Koch's postulates. He discovered that the disease anthrax is caused by the bacterium Bacillus anthracis, that cholera was caused by common marine bacteria, Vibrio cholera, and that tuberculosis is caused by the bacteria Mycobacterium tuberculosis. So that's chapter 1.1, looking at our learning objectives. So let's see how we did. Which of the following was when humans first suspected the existence of microbes? Within the last 500 years, in the last century, in prehistoric times, or during the Industrial Revolution? Well, we made great strides in each of those eras to understand more and more. Those prehistoric times, Etsy using fungus to try to cure parasitic disease, and the ancient Chinese. Fermentation by yeast produces what two products? Blank gas. So alcohol or ethanol. Ethanol is the type of alcohol produced, and carbon dioxide gas. That is correct. And who is considered the father of Western medicine? Antony van Leeuwenhoek, Hippocrates, Louis Pasteur, or Robert Koch? Hippocrates was our father of Western medicine. Which of the following is not true about the process of fermentation? It's used to make foods such as cheese and bread. It's carried out exclusively by unicellular eukaryotes, such as yeast. We'll get to our breakdown of different cell types in section that's coming up in section 1.3. It converts sugars to organic acids, alcohols, and or gases such as carbon dioxide. They can help preserve foods and prevent spoilage. If you said B, you are correct. There are other single-celled organisms and other organisms that can carry out fermentation, bacteria primarily. Which individual first and animal cells can also do that, like our own cells produce lactic acid. Which individual first observed unicellular organisms, which he called animalcules, using a microscope that he developed? Van Leeuwenhoek, Thucydides, Louis Pasteur, or Robert Koch? Nope. Those are supposed to be. PowerPoint skills failed there. All right, so that one, that's Antony van Leeuwenhoek. All right, so a double check on our objectives. At this point, everyone should be able to describe how our ancestors improved food with the use of invisible microbes. Describe how the causes of sickness and disease were explained in ancient times prior to the invention of microscopes. And describe some key historical events associated with the birth of microbiology, including the contributions by Van Leeuwenhoek, Louis Pasteur, and Robert Koch. Right. That's it for this section. So now you should take a look at the discussion questions, see what ones you can answer. Take a look at the PowerPoint slides on your own. Click on a few of those links. If you need some additional information, take a look at the supplemental videos that are in our D2L shell under the content for this chapter. And take a break before you come back for the video on chapter 1.2, the systematic approach. All right, I will see you in the next video.